Hello, everyone. Thank you for giving me the chance to talk today. So my name is Ciara. Like Kieran said, I'm supervised by Juan and Dario. And today I'm talking about a project that I've been doing with Dr. Xingcheng Yang and Dr. Vishal Raoji, as well as Laura and Joyner. And this is on the design of a non-invasive motor neuron interface for multidimensional control in tetraplegic spinal cord injury. So tetraplegic spinal cord injury is a debilitating condition. It affects around 20 million people worldwide, and it's associated with significant disability and loss of independence. Particularly in spinal cord injury, trauma to the vertebrae of the spine can lead to a lack of communication from the brain to muscle, such that you lose function of your limbs, depending on the position of the injury. Particular interest, restoring hand function is important to patients with spinal cord injury because this is a very dexterous part of our body and is responsible for performing most daily functions such as messaging, cooking, cleaning, reaching, grasping, etc. So people have been looking at trying to design a neural interface that takes some of the spared activity that exists after spinal cord injury. So it still resides in the spine. There are spared motor neurons, and trying to use this in conjunction with recording with electromyographic techniques to translate the spiking activities of these motor units into online control paradigms whereby you can interact with digital interfaces and try and improve the quality of everyday life. So some work that has been moving towards this has been recording with high density surface EMG the intended movement of people with spinal cord injury. So they will imagine the movement they want to perform, and we will pick up the spared motor neuron activity and decompose it with typical blind cell separation methods, which I'm sure lots of people here are aware of. And slowly people are now translating this into online control. As you can see here, that someone who has kind of complete paralysis of the upper limb is using the firing rate of a single or cumulative activity of a pool of motor units to track a rectangular or trapezoidal profile. And you can see in real time the spiking activity being decoded. But the problem so far is that this hasn't necessarily translated into practical applications, whereby we can actually harness this to do everyday tasks, such as navigating a screen with a cursor and click. So we wanted to move a bit more towards multi-dimensional control using motor unit activity, but also see if they are able to improve the performance of control with these motor units that we can record using EMG. So we designed a neural interface using free high-density surface EMG grids, and then we reverse engineer the activity of the raw EMG signal using decomposition to obtain the spared motor unit spiking activity. And we then map this into an online neural interface, which we progressively increase the dimensionality of the control. So from 1 degree freedom tasks using a single motor unit to 2 degree freedom and even 3 degree freedom tasks. So with this, we ask kind of 3 main questions. The first is if we can use spared motor unit activity in spinal cord injury to form controllable degrees of freedom in a neural interface. Secondly, if we are able to harness these spared motor units, can they use them in a continuous control manner? Is that control intuitive, and is it improvable with practice across sessions? And finally, what is the effective dimensionality of the control? Can we extend it to multiple degrees of freedom? So for this study, we recruited 3 participants. These participants all suffered from a cervical spinal cord injury, meaning that they had paralysis from the neck down. Some had some limited residual movements left. Most of these people had the injury around a year or 2 before they engaged in the study, and engaged in the study from kind of 3 to 9 months, depending on the time in which we recruited them. So the first thing we did was just see whether or not, if we can take the recorded activity from surface EMG, can we decompose it and distinguish the spared motor unit activity? So to do this, they were asked to track trapezoidal profiles where they imagined certain actions that they were trying to perform at maximum intensity. So they would see online the RMS of the EMG that we were recording. We took that signal and we decomposed it with typical blind source separation methods. And the important thing with this is that we can really partition out tonic activity that primarily derives from spasticity from the motor unit activity that is volitionally controllable. And this volitionally controllable activity is of primary interest to us because it can be used in the design of a neural interface. So once we have that motor unit activity, we then began 5 sessions. So they basically came to the lab for 5 sessions and we asked them online to track different profile shapes, which I'll show in a second. And they were asked to use with 3 different types of signals. The first was the RMS of the raw EMG. The second was with the activity or the firing rate of a single motor unit. And the final was with the cumulative spiking activity of all the volitionally controllable units. So they were asked to use the 3 different types of profiles across 5 sessions. So one was trapezoidal profiles, which we were interested in whether they can perform 1D proportional control, so they can hold a certain amplitude of control. The second was a sinusoidal profile, whereby they are asked to hold a certain level of control and then have small modulations around the amplitude. And finally, pseudosoidal profiles, which again is testing amplitude modulation, but also responses to changes in frequency. And this is just an example of some of the tracking they did with cumulative spike trains of all of the volitionally controllable units we decomposed. And you can see from kind of the second session to the last session, there is some improvements in performance. So here I have an example of one of the trackings of the participant. So they will track a pseudosynal profile. The bit at the start is a problem with the quadricento. If you've used the quadricento, you know this is common. So whilst that's showing, I'll explain a bit on the left. So across the 5 sessions, we basically calculated the Euclidean distance between the profile they tracked and the target profile that we presented to them. And you can see that there are some slow but steady improvements in performance with the different signal modalities across the 5 different sessions. EMG tended to perform slightly worse in the first 2 participants. In the last participant, the performance of EMG was relatively similar to that of using a single motor unit or cumulative spike train. We're not like 100% sure why, but it might be because of differences in the degrees of spasticity between participants. Because the nice thing about using a neural signal as opposed to just EMG is that we can remove the effects of spasticity or the tonic units. So once we had this 1D control and we were pretty happy with how successful they were doing in the task, we want to see, can we push this to 2-dimensional control? So can we effectively scale the task and can it be intuitive? So we designed this quadrant task in which the 2 axes were parameterized by the firing rates of 2 independently controllable motor neurons. So these motor neurons were typically from 2 different grids and associated with 2 different actions. So for instance, it might be extension of the thumb or abduction of the wrist. So they were using this to modulate the firing rates of the 2 motor neurons. And they were asked to basically move the cursor, so this circular marker, to different targets and hold it there for a small period to kind of emulate what we would do in normal control, like a cursor and click task. And once they held it for around 400 milliseconds, they would see a flashing green light and they move on to the next target. Again, the success rate was very good. But we noticed in particular that for the targets that kind of sat along the diagonal, whereby they were asked to co-activate 2 motor units, it was more difficult for them to achieve this task. And for us, we were wondering why. So we had a little bit of a closer look at some of the trajectories that they were taking with the firings of these 2 motor units. And you can see, for instance, if you look at some of the targets that sit on only one axis, they take a pretty straightforward, quick path to reach the target, so they can quite easily independently activate one unit and keep the other one switched off. But in the top left, if you have a target which requires high co-activation of both, they kind of struggle to reach the target. And this is kind of for a mix of reasons. One is it's quite hard for them to imagine modulating the intensity of both when they don't really see the end effector response. So they take this technique typically where they do a sequential approach whereby they switch on one unit, overcompensate a little bit, and then kind of introduce the other one and struggle to hold both intensities. So they kind of loop back a bit to the target. So we also gamified this just to make it more interesting and have a practical application. So this is an example of one of the participants performing a snake, whereby one motor neuron was controlling anti-clockwise rotation of the snake, and another one was controlling clockwise rotation. Okay, so yeah, we were pretty happy again that we could see that they can perform 2-dimensional control tasks. But it would be really nice if we could extend this to 3-dimensional, 4-dimensional control, because this would become closer to practical translation. So for instance, if you were using your laptop, you could have one motor neuron moving along the horizontal, one moving along the vertical, and then a final motor neuron could be used to click and navigate the screen or do what you want to do on the screen. So this is kind of moving it towards something that's genuinely useful for these patients. So we looked at 3 motor neurons in a pairwise manner in this quadrant task, and again wanted to see if they were independently controllable, which we did. So we thought, okay, let's design a 3-dimensional task to test whether they can do this in multiple combinations, so modulating the firing rates in lots of different combinations independently and co-activating. So again, this is an example of one of the participants using 3-dimensional control. So each motor neuron again was from a different grid and typically attributed to a particular action. I'll move the video along a bit. But you can see that there is some like issues where sometimes they can't completely hold for a long time one motor unit at a particular level. But a lot of the time they are able to independently activate these motor units. But again, the same problem arose where it was kind of due to co-activation that they struggled to kind of reach the target in the designated time. So example here on the top right. Okay, so we did all of these multi-dimensional tasks, but what I said at the beginning is the first 5 seconds were on first 5 sessions were on 1-dimensional control. So you might be wondering, with the second and third dimensions, which we didn't necessarily train, how did we understand that that control was actually intuitive for the other motor neurons we didn't use? So we had a look back at 1-dimensional control, and again asked them to track for the second and third dimensions, so the second and third motor neurons in these tasks. They were asked to track the profiles, and we had a look at the performance. So we call this kind of the untrained dimension, or the untrained motor neuron, because it wasn't used in 5 sessions at the start. And we just compared the performance outcome to the neuron that was used during the training. And we kind of see again that the profiles can be tracked quite well, and that the difference between the accuracy of the untrained motor neurons versus those that were trained is very similar, whereby the bounds of the trained motor neurons is shown in shaded gray, and the sessions in which we use the untrained motor neuron is just shown as the plotted points. So they have a large degree of similarity, which tells us that realistically we can pick any kind of volitionally controllable unit and the control will remain intuitive. And as a sanity check in the 2-dimensional task, we looked at the time of completion on the 2 axes of control to reach targets requiring the first motor neuron and the second motor neuron, where one was used in training and one wasn't. And we find that the time of completion is relatively similar. Okay, so we did a lot of other controls and very happy to talk about them in questions. But now, just to wrap up, I'll talk about some of the limitations of the study. So the first thing is that, as I said before, one motor unit was typically derived from one grid, which makes it relatively difficult for us to know how to optimize the setup for the design of a neural interface, where exactly these grids should be placed and how many channels do we really need to perform multidimensional control. The second is spasticity, which presents differently in lots of different patients. So primarily that spasticity can be dynamic. It's not necessarily we take a decomposition, we have a set of tonic units, and they always remain tonic. In tasks where they develop fatigue over several hours, some units can become tonic during the task, and then we have this difficulty kind of recalibrating, rechoosing units for the task, which we don't yet have a solution for, but would be a next step. And this leads to kind of the problem of generalization across the demographic of spinal cord injury. Spinal cord injury kind of presents itself in a very broad sense. So one of the participants, for instance, needed to do what we call pressure relief, whereby they're on the wheelchair and they kind of have to bow down to release pressure from their trunk. And doing that kind of changes their posture once they finish the pressure relief. And these changes in posture can also affect the performance of the decomposition and the tracking. So these are things that are patient specific. And again, we haven't yet found a solution to accommodate for these things. And the final point I wanted to make is translation between offline to online paradigms in spinal cord injury and in general in lots of patient populations is not very straightforward, because if you take an offline decomposition and look at it across actions and try and find independent motor neurons, they are not necessarily independent offline and online. You have to check always online that they remain independent. So it's not easy to translate and find independent motor neurons simply from just looking at offline data. So to summarize, in the study, we firstly confirmed, as with previous studies, that spared motor unit activity in spinal cord injury can be identified non-invasively. Secondly, that in most cases across the sessions in one-dimensional control, that motor unit decomposition would enhance control signal fidelity so that the cumulative spiking trains and the single motor unit tracking often was better than just using raw RMS EMG. And finally, that multi-dimensional control is achievable, so we can use up to 3 motor units in these control tasks, but challenges remain with co-contraction of different actions, and that kind of remains an issue to resolve, which we didn't really see an improvement with just repeating the task and asking them to train per se on the task. So we've also been doing this as an intramuscular approach, which is on the poster outside. So if you're interested in that, also have a look and feel free to ask questions. So we just wanted to see if it's possible to kind of target specific muscles using thin films and kind of repeat the same process of one-dimensional and two-dimensional control. We're kind of still in the process of this study, but happy to talk about it. And that's about it. So thank you very much for listening. I want to say thank you to everyone who's been working on the study, particularly Xing Cheng and Vish. Thank you to the people who funded the study. Thank you to Hillingdon Hospital, who facilitated a lot of the experiments that we did, and the physiotherapists who helped out. And most importantly, the participants who gave up their time to help us perform the study, who've been very patient and very kind with us. So thank you.